So I'm going to be talking to you this morning, without my mask, on the genetics of major depressive disorder. And one major theme of what I'm going to be telling you is to point out the work many people in this audience who've contributed to these ideas, so I'm not taking responsibility for any of the wonderful insights I'll be presenting to you today. And I'm just going to begin by just giving you a little glimpse of where we are in terms of genome-wide association studies. I'm assuming you're all familiar with this type of presentation, from which it's very clear that there are large numbers of peaks exceeding an accepted genome-wide significance threshold. This is for a study published in 2021. There's another one most recently now with a very large number, more than 1.2 million individuals recruited to identify more than 100 loci that contribute to the risk of this disorder. And what we've learnt from these studies is what we've seen from every other study of complex trait conditions, that this is a highly polygenic disorder. I'm just plotting out here the odds ratios for the loci that have been detected, and you can see that they're really small, with a mean of about 1.03. This, for those of you who know this literature, is relatively small. So for schizophrenia, the similar figure is about 1.06, and there may be some stories around this, but it just contributes to the fact that what we're dealing with is a very polygenic condition. But that picture I've just portrayed of the successful mapping, highly polygenic, and I put it in the context of other conditions, might lead people to think that this is similar in many ways to what we're finding with schizophrenia, bipolar disorder, obesity, hypertension. But there are a couple of features that I think need to be emphasized that particularly mark out work that's been carried out with depression. And the first of these is that this really is primarily an environmental condition. Less than half the risk is genetic. And unlike many disorders where we say there is an environmental risk and we have no idea what that is, for this disorder we do. We know that stressful life events, bad things happening to you, you all know this, they make you miserable. And although we don't have that robust causal data carried out in laboratories to demonstrate this, there's sufficient epidemiology, longitudinal studies to indicate that when bad things happen, the risk of depression goes up. And with certain conditions, that's a very substantial risk indeed. So this is unusual. We don't have this handled for many other conditions. And the second thing that marks out depression as a condition to look at is that it's very heterogeneous. And it's heterogeneous in many different ways, and I'll be illustrating this in the rest of what I've got to say. But just think of it yourself as like this, that I can make a diagnosis of someone with depression who has been bedridden for weeks, who's thought of killing themselves endlessly, who has resisted all treatment that I may have given, including electroconvulsive therapy, an arsenal of pharmacology, and with a recurrent condition that's gone on for the whole of their life. That would be a diagnosis of depression. But similarly, someone who may have had a relatively minor mishap, something like getting their paper rejected, I don't know, something you might take seriously. They get depressed for 2 weeks, they lose sleep, they wake up early. They also would meet criteria for depression. And both of those cases will get lumped together under the same diagnostic category. And that should tell you already that there's something slightly odd about the way we deal with depression. But as I'll tell you, there's even more than that. So if you just look here, these are the criteria by which I'm trained as a psychiatrist to make the diagnosis. On the left-hand side is the US, the DSM-based, and on the right, the European International Classification of Diseases. And there are a couple of things to note about this. The first is, of course, that the two sides don't quite agree. So there's 6 symptoms on the right, there's 5 symptoms on the left, but they more or less do, if you read through. There's a sufficient agreement in what they're saying. But note also that both of them have this sort of laundry list approach, that you could have one and then the other and then 2 of this and so on. There's a number of different ways in which you can meet the criterion for having major depressive disorder. It's not like you've got to have A, B, and C. It's not like that. There's a list of these things, and you can pick and choose. And that leads to one of the more extraordinary features about this. So this paper here worked out the number of ways in which you could become, which you could meet criteria, and it's something like 1,400 if you add up all the possible ways in which you could meet criteria. So that's a little strange. And the other thing to point out, which you can see from the list I've just shown you, but I'll emphasize in the next slide, is that you can actually meet criteria by having totally opposite symptomatic criteria. So when I'm asking you about whether you're depressed, I'll be asking you about whether you sleep too much or whether you sleep too little, whether you eat too much or whether you eat too little. That's weird. That really doesn't make sense. Now, the reason we have this is because the criteria have been devised to ensure, guarantee consistency between examiners, between psychiatrists, between trade interviewers. And it's a syndrome. We don't really understand how these things go together. So we sort of at the start know that the pattern is not necessarily based on a biological single entity. So you know a little bit about where we are in terms of the genetics, and you know a little bit about the problems I've got in terms of the diagnosis, and I've mentioned to you about heterogeneity. So what I'm going to spend the rest of the talk doing is looking in a little bit more detail about what the genetic analysis carried out so far might tell us about this condition. And I'm not going to be talking about the genes. I have a bit of a prejudice here, I'll admit this. I don't think we have found any genes. There's an awful lot of chat about the location of genes being near loci and whether there's some transcriptional evidence. I'll put that all to one side and I'll basically tell you it's garbage. I mean, you can argue with that, but I don't believe any of that. We're not going to talk about it, at least I'm not. So what I'm going to be talking about more is what we've learnt from the genetic analysis carried out with the SNPs in the sorts of data that I've just shown you from the GWAS studies. And the first question I'm going to ask, a very simple one, how many of the established loci are reproducible in ancestry match studies? Now, it's rather surprising that this question doesn't get asked very much. And if you ask this of any condition, I think you'll find difficulty getting a literature that would really address this. And those of you interested in developing some method to identify what's going on, please go ahead, because I think there's a gap in the literature to deal with this. There is one paper I found, this one here, which was looking in East Asian, but also looked at other ancestries, and specifically worked out what they expected the reproducibility rate would be given the effect sizes. Now, I'm sure you're all familiar with the fact that when you carry out one of these studies, if you find something, the effect sizes are typically overestimated in a first study, that you can't rely on that. You have to deal with a whole set of confounds that might deal with this. I know Eliezer has thought quite hard about this problem of reproducibility. So I'm not saying these people get this right, but at least they make an attempt at the answer. And what they said was, well, we assess evidence for reproducibility, we work out what we think it should be, and the answer they get was that for a reproducibility rate of 84%, they found compared to an expected rate of about 100%. So a substantial and indeed significant reduction. So why might that be? What does that indicate? Is this a failure of the actual methodology? Is it a failure of the way that the data sets have been collected? Is it something to do with the condition itself? And as you probably guessed from what I've said earlier, I think a lot of this will turn out to be do with the way that we make the diagnosis and what we think is the underlying biology. But let's pursue this a bit more in detail. So this is a summary that I've made of all of the genome-wide association studies from 2009 up until today. And if you concentrate on the right-hand side of the table, I've shown loci, and you can see the number of loci discovered is resolutely 0, 0, 0, down till about 2016, when suddenly it begins to go up. And the reason for that is because of the way that the diagnoses were changed. And the key paper in this respect is this paper that came out from the 23andMe, the Consumer Genetics Consortium, where they for the first time discovered more than 10 loci that exceeded the significance threshold, as you can see in this plot, and were able to replicate them because they had access to a large sample. Now, what they did in making the diagnosis was apply a relatively simple criteria. So although this slide looks busy and there's lots of points on it, what it resolves into is simply a single question: have you ever been diagnosed with clinical depression? There are different ways that's got asked, but essentially it's a single item. And if you said yes in one of those varieties, you'd be counted as a case. And that's very easy to get. I can collect data from all of you in this room in about a few minutes. We could just put something up on the web, you could click a button, and I'd know. And because of that, I can get a very large sample size. So this is the numbers that they got, 75,000 cases, and look at the replication size, it's pretty good as well. It's a very large sample, and that gives them the power to detect effects. And then this is sort of well known, very standard. And you can draw really from that data I showed you in the previous table, a plot that shows you, as you increase the sample size, which is in this case shown on the horizontal axis, that the number of loci that you detect at genome-wide significance increases. So all that's well and good. And you can see why they did it and why we should continue doing it. But let's just go back a moment. So I showed you this, and you look through those criteria, and probably some of you wondered whether you'd had it or not. And as you read through those criteria, some of them are obvious. Well, depressed mood, I think I know what that is. Insomnia or excessive sleep, that's pretty obvious. But what do I really mean by diminished concentration or indecisiveness? I mean, I'm pretty indecisive most of the time. Am I really going to meet this when I'm sort of going to qualify as a criterion for depression? And these recurrent thoughts of death and suicide, well, I have those most of the time. I'm sure most of you do. Life is miserable. So how do I decide when I meet somebody whether you really meet those criteria? It's not so easy. It needs a clinical interview. It needs somebody trained. And just getting this information from a single item is not going to do the job. So what do we know about these short screening tests? They don't do really well. So about half of the results from a single item test, like the ones I've shown you, are false positives. And even 2 or 3 item tests don't really do much better. This is a well worked over field. I'm summarizing in a single slide a very large literature. It just comes down to this: they don't work very well, and we'd expect half of them to be wrong. Does it matter? Maybe it doesn't. There are some people who think it doesn't. This slide comes from a review in Neuron from my colleagues, who argue that if you look at the self-declared depression, you'll find an overlap with major depressive disorder in this simple sort of Venn diagram form. And what we're really interested in is that purple blob in the middle, and we'll capture it. That's sort of the argument that they make, which seems reasonable, I think, on the face of it, except there is the possibility that maybe what we're really interested in, all that core problem about sleep and not being able to eat and your really suicidal mood, maybe that lies on the left of that figure, in that sort of teal-coloured lunar-shaped thing on the left. Maybe that's what's there. We don't really know. And this is where I'm very grateful to Na, who's sitting in this audience, because she has, I think, a very nice demonstration of why, in fact, looking at the central purple blob might not be the right place. And I'll show you how she thinks about this. So I'm showing here a number of loci. Each little red line represents an effect size from a locus, a risk locus for depression, diagnosed by this single item. I've been diagnosed with depression. And the horizontal axis is the effect expressed as an odds ratio, which means if it's 1, there's no effect. And the little horizontal lines represent the confidence intervals to show you that it's significant. And as you'd expect, they're all significant. So now we're going to look at the same loci, but diagnosed in a more systematic clinical way for a DSM diagnosis. And note a couple of things. The first thing to note is that not all of them are now significant. The effect sizes are generally smaller, which is sort of what you'd expect because we're not looking at quite the same thing. But note, most importantly, that the pattern of effects is the same. If you run your eye down that line that's formed by the dots for each of those effects, you'll see that it's the same in both. So that would tell you that, yes, this would agree with what that review I showed you suggested, that there is an overlap and we can find the same things. That seems reasonable. But Bernard goes one step further. She then asks, well, what do we see for other conditions which are not related to depression? So the next one up is neuroticism. Neuroticism is a personality trait. Neuroticism is not depression. There are many people who get neurotic. They're just, anxious people. They worry about tiny things like getting their papers published and getting a job, miserable stuff like that. That's a personality trait. That is not depression. And then schizophrenia, that's not depression. Hallucinations and delusions, nope. Those are exclusion criteria. But if you look at the pattern of findings there, they're very similar. And smoking, that's not depression. But the pattern again is the same. So what that indicates is whatever those loci are, although they are occurring in depression, they're not specific to the condition. So that's an indication that if we were chasing these rather poorly defined phenotypes, we're not going to be getting what we really want. So there's a problem about diagnostic heterogeneity. And I've hinted also, and I gave you one example, just a clinical example, about clinical heterogeneity. That's to say that we might be dealing with different conditions. And when I say different conditions, it's not like these are well-contained entities. It's not like we've got a depression which is clearly defined due to A and another one clearly defined and due to B. It isn't like that, but it's probably a messy mixture of things. It's certainly not just one big homogeneous condition. That seems to be pretty certain. But exactly where the boundaries are, that we don't know yet. How can we begin to parse this and get some insight into it? Well, I've told you already that the major effect is not genetics, it's the environment. And that gives us a handle on trying to pull this condition apart. And the environmental effects, as they interact with depression, has become a sort of an industry. And I won't go into the rather sorry history around this, but just to put into your mind two ways that you can think about how we might detect the interaction between these two things. So on the left, I put a little stick figure to show you how we think of the relationship between depression and a stressful life event. There's a fixed interval, it's about 3 months, between the time when a bad thing happens to you and you have an episode of depression. And if I collect data like that, I can then ask whether that effect, the chance of you becoming depressed or not, is dependent to some extent on your genetic susceptibility. We know that about half the people who have an episode of a stressful life event will have a change in their mood which might qualify as depression. So if I get a group of people with depression, then maybe half of them I can see some indication that it's due to this. So we know it's not like one-to-one. Most of us know this. There has to be something else going on. So on the right-hand side of this figure, I've shown two ways in which we may see this. One would be in this change in the genetic risk according to the level of environmental risk. So the horizontal axis shows the level of environmental risks, bad things, the worse they are, happening to you on the right, and the total liability, both genetic and environmental, shown on the left. And you can see that there's a splaying out of the genetic risk. Those with high genetic risk show an increase above and beyond what you'd expect from the low genetic risk. It's not an additive effect. And then below, I've shown the possibility that you might see a change in the heritability. In a simple sense, you might think that people who are genetically loaded won't take so much environmental insult to push them into being depressed. So you might expect that those people who report not exposed to adversity would have higher heritability than those who report that they had a bad thing happening to them. That's a simple way of thinking about it. There are others, but we might expect some change in the heritability. So what do we find from the literature? What do people report? So here's a very simple illustration that matches directly what I showed you before, that there is an increase in the risk of depression by exposure to a traumatic event. This is from a Dutch group, and they use a polygenic risk score to get at the genetic susceptibility. That's plotted on the horizontal axis, and show that shows a change in the likelihood of depression depending on whether there's a presence or absence of childhood trauma. So that fits that first model I showed you. Not everyone finds this. So very different figure. Same plot, polygenic risk score on the horizontal axis, probability on the vertical axis, and they're looking at environmental adversity across a large group of people here, and you can, don't need me to point out to you that plot looks completely different from the one I've shown before. And a third report doing the same thing, trying to look at trauma on a risk of depression. This group has a very large table at the end of their paper which summarizes, and you really need to concentrate only on the right-hand side, which is the p-value there, and you can see there's really nothing significant in their interaction between the genetic risk, that's again indexed by the polygenic risk score, and childhood trauma. So people are disagreeing. We're not really seeing any consistency yet. What about the other component, the change in the heritability? So here's one result from the UK Biobank, where they were looking at a change in the SNP-based measures of heritability on those reporting trauma. So it's 24% if you report a bad thing, and only 12% if you don't report trauma. So looks interesting. It's the other way around from the one I suggested to you, but they have some confidence intervals. It's sort of got a p-value and so on, looks robust. But Andy Dahl, who might be sitting in the audience, carried out an analysis, much more sophisticated, I think, on the data that we provided with him and came to a very different conclusion about this. So at the moment, this is a mess. We don't really know what's going on. We need people with computational skills to think about this, and also people like myself to give you better, more complete data sets. So I've talked a bit about the clinical heterogeneity and how we might access that through understanding how environmental risks operate. And I want to now turn to a slightly different issue, but again indexed from what we're getting out of the genetics, which is what I'll call a cross-disorder similarity. And I'll introduce this with another table summarizing the literature. So I'm showing genetic correlations between depression on the left and a number of different traits, and I'm giving you the measures estimated from both twin studies, that's where it says RG twin, and then from GWAS studies where it says RG SNP. And two things to note from this. The first is that the estimates from the twin studies and the SNP basin are not the same. There's sometimes really quite marked differences between the two. So if you look at the bottom, when we're looking at the relationship between that personality trait I mentioned to you before, neuroticism and depression, we're getting estimates from twin studies of about 40%, whereas the SNP-based ones go up to about 74%. But note that there's a big variation in that. The second thing to note, which you won't necessarily know unless you've been trained as a psychiatrist, is that you're not supposed to see any correlation between major depressive disorder and, say, autism spectrum disorder. I mean, I took an exam when it said, is there a correlation? If you ticked yes, you'd fail. It's like there wasn't one. Now, SNP-based analysis is saying there is one. What's going on? Who's right here? The geneticists? What do they know about psychiatry? Or the psychiatrists? So this is a problem in the field, and how we understand this, I think, has some interesting implications about how these analyses were carried out, and also about how we come to make these diagnoses in the first place, an issue which I've already mentioned to you. So I'm going to turn briefly to discuss this issue with just one example, and I'm going to show you some work that Richard Border has done, attributing, to some extent, maybe all of the correlation, being attributable to assortative mating. So what is assortative mating? Assortative mating is when you choose somebody who's a bit like yourself. They might look like yourself. They might have some of the attributes that you share. And most of us are familiar with this. We don't imagine that we're going to choose to spend the rest of our lives with somebody we have nothing in common with. Most of us don't think that's going to happen. So there are three genetic consequences of assortative mating. The first is there should be an increase in the genetic variance in the population. The second, there should be an increase in the correlation between relatives. And lastly, there should be an increase in homozygosity, measured as a deviation from the Hardy-Weinberg equilibrium, particularly at those loci that contribute to the trait we're interested in. And one way of thinking about this, which we can thank my colleague Peter Vischer for, is a gametic phase disequilibrium. So let me just talk you through what we mean. So imagine we just look at the odd chromosomes. Let's say what I've shown there are chromosomes 1, 3, and 5. And we have trait increasing alleles in orange and trait decreasing in blue. And I can score across each chromosome what the number of alleles are that contribute to the trait. So the chromosome 1 in this case has 5 increasing alleles and so on. Now, under random mating, we should find that there is no correlation with the even chromosomes. So I'm now portraying chromosomes 2, 4, and 6. Chromosome 2 happens to have 1 increasing allele. Chromosome 2 has 8, chromosome 4 has 8, and chromosome 6 has 1, and so on. So the number of trait increasing alleles on the odd chromosomes does not predict the number on the even chromosomes. I think that should be obvious if you just apply simple segregation to chromosomes. Nothing unusual there. But in the presence of assortative mating, that assumption breaks down. And in fact, you will find that the number of trait increasing alleles on the odd chromosomes will predict the number of trait increasing alleles on the even chromosomes. Simply because we're no longer randomly selecting from the population. We're finding people that are like us. And that, of course, now provides us with a simple test of the presence of assortative mating, that we can use this measure of this relationship, the breakdown of prediction. So Peter had got that far. What Richard has done was consider whether you could argue also for assortative mating on multiple traits, not just 1, but 2 or 3 or 4. This little picture is just to show, if you're going to marry a beautiful woman, she also has a jet or something. I don't quite know why. So Richard has this really nice paper. Very happy to have worked with him on this to indicate that when you look at cross-trait assortative mating, so now we're dealing with exactly the same thing, but now we're going between traits, not the same trait. It's widespread, and it inflates the genetic correlation estimates. So the simple argument is the linkage disequilibrium arises between trait-increasing alleles for traits Y and for Z, and then they will appear to be genetically correlated. They're not. It's just the assortative mating that makes them look so. And I'm just summarizing his results here. He suggests that a substantial fraction of the effect correlation estimates among many pairs of psychiatric traits are largely consistent with assortative mating. That's the XAM acronym there. Modest levels of diagnostic error, when coupled with the assortative mating, can fully account for some previously published estimates. And I'll show you a figure from his paper, which is this one here. It's a little bit hard to understand, but I'll try and talk you through it. So he has this value gamma, which you can see on the bottom, which is essentially the ratio of the assortative mating to the empirical observation. So if there's 1, it's spot on, and if it's either side, it's not the same. We're really looking at deviations from that. And what he's looking at is what happens if you have 1 generation of assortative mating. That's where it says generations of XAM1, 3, and 5. And then he's put some values for the misdiagnosis rate. And what I'd be interested in, for example, is what I told you before, that is there a relationship, for example, between autism and depression, or in this case, I think he's showing schizophrenia and depression. And you can see that certainly with up to generations of 5 generations of assortative mating, that the green bars are well shifted beyond the value of 1, which is consistent with what I told you before that he summarized. Notice that his misdiagnosis rates here are sort of 5 to, the maximum he shows is 15%. Well, remember what I told you before. The cases of depression diagnosed by a single item, about half of them are incorrect. So we could certainly attribute a large proportion of what's going on in the literature to a combination of misdiagnosis and assortative mating. So what's to be done about this? I'm going to end my talk with just talking about 3 things, how we might progress. One thing clearly is that we need better samples. We need to give people working in this area, computational people like yourselves, we need to give you better phenotypes and diverse ancestry. It's become a sort of sine qua non now in this field. And I'll talk about two other things, digital phenotyping and phenotypic imputation. Now, just briefly to deal with the first thing about more samples, this is hard work. It means I have to train people, I have to get them to collect data, I have to check the quality of the data. It might take an hour sitting down with one of our cases to really get a good diagnosis and all the related features, because I want to make sure that it's depression that's not related to smoking or alcohol or drug abuse. I want to make sure it's depression that's come out of the blue or not come out of the blue. I need all of that information. It's hard work to get this. So it's going to be slow. It's not going to happen quickly. The second thing I put down here, digital phenotyping might help us in speeding things up. What I mean by digital phenotyping is the use of devices like your phone, maybe a watch, maybe some other wearable, which continuously collects data on you, which I can then relate, but again I'd need a validated assessment, which I can then relate to your mood. And when I've done that, I should be able to find those features on your phone which predict your mood accurately. And we're not the only people doing this. There's a large group. I'm just showing you very briefly some data. This is collected from UCLA students who were asked to put an app on their phone to collect their activity. This, as you can see, has been a longitudinal study. A horizontal scale here is in years. And some people have been giving us data for indeed, up to a year or more. And we've been trying to extract that data, make sense of it in terms of what their mood will be. And I'll just give you one example of how this might work. What we're seeing here is taking on-off time for a phone to indicate hours of sleep over a 3-month period. So the estimated hours of sleep are shown at the top here. And you can see there's a little block of a few weeks where somebody's beginning to wake up much earlier than normal. And this is related to their depression score, which you can see beginning to creep up during this period, as you'd expect from what I told you, being given that changes in sleep are indeed an indication of depression. Not perfect, but it's an indication of what's going on here. So it's likely to produce something. We may improve things. Early days yet, I would say. Not that early. I found this. It's just been published. There's a review article, now like 200 or 300 papers in this field. There's definitely a lot going to come out. Early in the sense we don't really know what it's going to contribute, but definitely an area to watch. Complicated data fields. Those of you who love running artificial intelligence, neural nets, whatever, please come and work in this field. There's tons of data for you to play with, and we'd really like to know what it means. The next thing I wanted to talk about briefly is phenotypic imputation. And this is some work that, again, Andy and Noah have been carrying out, so you'll know some of this already, but I just want to show you what I think are the really interesting results in this. So this is a data set from UK Biobank with a single measure of depression called lifetime major depression with a DSM diagnosis, and you can see that there's just one genome-wide association hit. And this was from a self-administered questionnaire. So not great, but it's better than many other things. But it's only on a very small proportion of UK Biobank. So the question was, what would happen if I take that data set and I can impute this same phenotype onto the full 500,000? And if you do that, things get a lot better. And that's, I think, pretty expected. It should be the case, but it's nice to see that demonstrated. But what is, I think, unusual, and it's really thanks to Andy and Na's insight into here, that we could do something else other than just increase the sample size of and get more GWAS hits, that we could distinguish specific and non-specific signals of depression in these self-report measures. And I think that's been a really important insight, given what I've told you before. And the way they've done this is to use soft impute to identify latent factors that cross-cut various depression phenotypes. And I'm just showing you one figure from this paper where you can see that we're pulling out basically factors, and think of it as a factor. And depending on what those factors do and where they're loaded, you can essentially residualize them and remove them, saying, I don't think this factor is related to what I'm interested in. This is not depression. We'll get rid of that one, remove it from the data. And if you do that, you go from this figure, self-report diagnosis, not interested, bad phenotype, everything I've told you before. Now we'll ask Nara and Andy to clean it up, and we get that. I think that's amazing. Very impressive piece of work. Thank you, Andy and Nar, for doing that. So what do we know? We know that we have diagnostic and clinical heterogeneity, and they confound the interpretation of the results. One interpretation of where we are at the moment is that the imposition of this simple phenotyping approach has really caused more problems than it's solved. Another interpretation is we now have all this great data and we need to understand it. Up to you which view you take. And the next thing is, what do we do to improve things? Well, we can identify etologically homogeneous types of depression using the genetic analysis of digital phenotypes, and we can try and distinguish the specific and non-effects using the sorts of approaches that I mentioned to you before. And I want to end with a final thought to you, which is that for those of you who don't know this book, I thoroughly recommend it, not just as an introduction to the commonest, one of the major causes of disease and mortality, but as a general understanding of how scientists and or clinicians have tackled diseases. And if you go back and read the descriptions of what people thought about cancer in the 1940s, you'll see there are many similarities to what I've been telling you today about how we think about depression. The only difference is the organ and the phenotypes. And that type of thinking, the clinical thinking, particularly joined with the types of really rigorous scientific approaches, some of which I've tried to illustrate to you today, is the way I think we can take this forward. And I'd like to thank a lot of people for this work. I have a group that we've been working with over the last couple of years. One of the good things about the pandemic, it's forced us to think about this hard. So thank you very much.